Right. First of all, for those who haven't started this lesson at your classes or schools, I'll give a small introduction how this lesson contributes for your final paper. Right. Now, if you see, you know that your final paper consists of 50 MCQs. Now, wait, 50 MCQs. So 50 MCQs means direct 50 marks, right? So from those 50 MCQs, every year, minimum 3 MCQs are questioned. 3 MCQs. So if you consider the part 2, structured essay almost carries one structured essay carries, if you see 5 marks, those five from the 5 marks, almost 2 marks can be two. I'm representing Q for a mark, right? So 2 marks will be carrying. So if you see essay will be carrying almost 3.75 and a half essay can be asked. So this almost and overall we can just take it as 3 marks. So total 8. So 8 marks, minimum 8 marks. 8 marks in your final paper. Now you might think just 8 marks, but this would worth for those who are aiming for a A, right? A for almost this plays a huge role for those who are aiming an A with a rank, right? Because most of the students does not pay much attention for this lesson because it is mostly theoretical lesson. So this plays a huge role, right? Almost 8 to 10 marks will be carried by this lesson from your final paper, right? Clear? Now, right. Right. Now let's go to the lesson. Now, this lesson mainly speaks about genetic materials, right? I'll give a frank idea on that. This will be just speaking about DNA, RNA, and proteins, and then it will be going further onto the various mechanisms, disorders, and it will be going to gene technology. So I'll just write what this lesson contains. Now, here, right. First topic today, which will be discussed is DNA packaging. DNA packaging. Second topic is DNA replication. Third topic is genes, operons, and then the fourth topic would be protein synthesis. Protein synthesis, right? And fifth topic would be mutations. And the last topic is gene technology. So this is what your unit 7 contains, right? Almost 1, 2, 3, 4, 5, 6. So almost 6 topics, right? These are the complete content under unit 7. Now, here, except for gene technology, all other topics are small content, right? So this complete lesson without excluding the gene technology almost carries excluding gene technology, it carries almost 60% of the lesson. This gene technology part only carries 40%, solo 40% of the lesson, right? Is that clear? All other portions except for gene technology will be carrying 60% of the lesson. Gene technology solo will be carrying 40% of the lesson. So it is a huge content part. So there we'll be mainly explaining, we'll be discussing about the applications, what we have learned so far, right? So we are not going further onto that. Let's go with the flow. So today we'll be starting DNA packaging. A simple part, we can finish it with today. And if we finish it, uh, for those who haven't started the lesson, if you want to take a small note down, take it. Just a small rough diagram, right? If you want, do you want few minutes to take it down, or shall I move it? Shall I move on? Guys, right, right. So now, right, now I gave you just rough idea on what this lesson contains. Now the topic today is DNA packaging. Before going to onto that, now we should know something that under this lesson we will be mainly studying about comparisons between eukaryotic cells and prokaryotic cells, right? What we'll be learning the content. If we learn under DNA packaging, we'll be comparing the DNA packaging process with prokaryotic cells, eukaryotic cells. If we learn under DNA replication, we'll be comparing between prokaryotic cell and eukaryotic cell, so there would be a comparison, right? Now, before starting, as I said initially, so let's start off with discoveries that have been discovered by the scientists. Now we know that a DNA molecule would be looking like this, right? I'm here. This is a DNA molecule. Now here, this diagram was the DNA molecule is a double stranded molecule. So this was proposed by two scientists, as I said. So first discovery under discoveries, discoveries. The first discovery. First one is DNA is double stranded. Double stranded. Who proposed that? Two scientists. First one was James Watson and Francis Crick. Right? These two proposed that a DNA is a double stranded molecule using some sort of a technology which is called X-ray crystallography. X-ray crystallography. Right? This X-ray crystallography was proposed or discovered by a scientist called Rosalind Franklin. Rosalind Franklin. Right? So under the first discovery, there are a few coming under, right? Now, if we take the next one, discovery is a scientist called T.H. Morgan and his group of companions. They said that a DNA, they said that chromosomes, we might have learned about chromosomes, some sort of knowledge under cell division of unit 2, right? He said that chromosomes are made up of two things. One is DNA and other one is proteins. Is that clear? These are the two discoveries, right? So the lesson starts off with these two discoveries. What are the discoveries? They said that first one, DNA is a double stranded molecule. This was said or proposed by two scientists named James Watson and Francis Crick. These two scientists used X-ray crystallography, which was proposed by Rosalind Franklin to propose that DNA is a double-stranded molecule. The second discovery is a scientist called T.H. Morgan and his group of companions. They carried out various experiments and they said that chromosomes are made up of DNA and proteins. Is that clear? These are the discoveries. Simple. Clear? Put up a thumbs up, Mark. Is it clear? Let's quickly do those. Is that clear? Right. Thanks for responding. Right. This is the rough idea, right? These are the discoveries. Now let's go to the direct topic, which is DNA packaging, right? Right. Now, before going on to the direct topic DNA packaging, they are introducing us to some sort of thing called architecture. Architecture of chromosomes. What is this? The name itself says something. It says that architecture of the chromosome means the arrangement of chromosomes, right? So under this lesson, initially they are saying that DNA, they are referring DNA to be chromosomes. Is that clear? They're not saying that DNA is chromosomes are DNA. They are saying, they are just, while mentioning, they are saying DNA of prokaryotes and eukaryotes are chromosomes. So students should not misunderstand that as chromosomes are DNA. We know from the discoveries just while ago, I said that scientist called T.H. Morgan and his group of scientists, they said that chromosomes are made up of DNA and proteins, right? Here, with the note, with the flow, they are saying that DNA of prokaryotes and eukaryotes are said as chromosomes. Don't confuse that. They are saying DNA and proteins are the ones considered to be chromosome. But with the note, they're just saying DNA of prokaryotes and eukaryotes are chromosomes, right? Right. Now, under architecture of the chromosome, we'll be understanding how the arrangement, they're giving a small note under that, how chromosomes are arranged. Now, as I said, when the lesson started, as I said, right. Now, if you see a prokaryotic cell, prokaryotic cell. Now, first, before we start off the lesson, we should know certain things, which means not new things. You might know this under Unit 3, but at that time, we might have just memorized them. So I'll just recall them. We know that prokaryotic cells have what type of DNA? Circular DNA, right? Circular DNA, right? As they have circular DNA under the a table, you might um, remember that a table was given under Unit 3. There was a table, a comparison between the prokaryotes and eukaryotes. So they said that a word called circular chromosome. Can you remember that? So they are referring circular DNA. So circular DNA is present means there are circular chromosomes too. So there's prokaryotes have circular chromosomes too. Circular chromosomes too, right? Second is prokaryotic cells does not does not have an organized nucleus. Now, this is the knowledge we have from our O levels. So, if you ask me, have they given this point in A levels? No, they haven't given this point in A levels. But indirectly, they are trying to convey that this is the knowledge that you have from OLs. OLs, they are saying that prokaryotic cells does not have an organized nucleus. Now, in A levels, they are saying that prokaryotic genetic material, the DNA, is present in a specific location called nucleoid, right? The word called nucleoid is introduced. Nucleoid. So, what is this nucleoid? Nucleoid means the location where the DNA is present in a prokaryotic cell, which means in cytosol, the specific location where the prokaryotic DNA that is present is called as nucleoid. So if you take a bacterial cell, now this is a bacterial cell. Now if you see in a bacterial cell, now the DNA is present in this manner, circular DNAs. Now see, this is the circular DNA. Now this is the cytosol. This is the cytosol, right? This is the cell wall. Now, what would you say for this part? Can anyone say that? For this part, the yellow colored part, what would you say for that? What is that? What is that? That is the thing that they are trying to say that is the nucleoid, right? This is nucleoid, right? Right? Nothing complex. They are saying that in a cytosol of a prokaryotic cell where the DNA is present. Now, if you see in a eukaryotic cell, the DNA is present organizedly in a nucleus. That is the difference. In a eukaryotic cell, it is present within a like a tent-like shape nucleus. But in prokaryotic cell, there isn't a nucleus. There isn't an organized nucleus. So the specific location where the DNA is present in a cytosol of prokaryotic cell is referred as nucleoid. This is not directly given under. This is not directly given in your uh, resource book. They are not. No. Now you have come to the latter part of the AL journey, right? So they won't be in the resource books. They won't be giving small ideas, each and every ideas. They won't be giving it. They won't be saying that nucleoid means that it is the location where the DNA is present in a prokaryotic cell. Each and every word they won't be giving definitions. They will be giving definitions only the specific parts which needs a derivation. Is that clear? Right. Now I'm introducing slowly for all the terms that will be discussed under this topic. Right. I have not yet gone for the process of DNA packaging. These are the words which will be going through. Right. Right. Now, right. Now, is that clear? This part? If this is clear, we can go to the DNA packaging part in prokaryotic cell. If this is clear. Is it clear? If this is clear, let me know. If it is not clear, please drop a message. I'll explain it again. Is it clear? Right. Good. Thank you for responding. Right. Then let's go for the topic directly. Right. Some might, no, your seniors might say that this lesson is a bit hard. It contains much amount of theory, but trust me, if you do your works properly and uh, maintain that recallingness and revise regularly, you might feel this is one of the easiest lesson, right? This has some sort of theory, but not complex. It is a very easy portion, right? Right. Now, let's go to the topic. So, a new topic, which is DNA packaging in prokaryotes. Oh, first of all, yeah, before starting this, wait. Before going on to the prokaryotic part, I'll introduce what they are trying to convey from the definition, right? Before learning a specific portion, we should know what they are trying to convey from the definition. Not just memorizing the definition, we should understand each and every word from the definition, right? Now, the definition DNA packaging, the resource book says it is the containment of genome. It is the containment of genome of prokaryote 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 in nucleoid. It is the containment of genome of prokaryote in nucleoid or nucleus in eukaryote. Can you understand something from this definition? Try to understand something from this definition. Now, if we break down this definition, now they are saying that it is the containment, right? Genome of prokaryote in nucleoid or in nucleus of eukaryote. Now, what is this genome? Genome means it is a collection of genes. Is it clear? Collection of genes. Now you might ask, what is genes? Now we have a rough idea what are genes. Genes means it is something we have within us. But to have a bit of clarity on that, I'll give us some sort of just a hint, which will be learned in future in this lesson. I'll just give a hint under that. Now, genes means remember it as DNA segments. What is this? Genes means they are DNA segments, not DNA alone DNA. They are in DNA segments. It might contain several DNA, right? Several DNA segments. So a collection of genome means a collection of genes. Genes means DNA segments, right? So they are from the definition, they are saying that DNA packaging is the process where the collection. Now think this is the collection, this is the genome, right? The green is the genome, right? Now this is the DNA. These are the DNAs, right? These DNAs are present, right? Now, if this genome is present, the process of having the genome, wait, I'll mention this green as genome. Now, this is the eukaryotic cell. What is this? Eukaryotic cell. In the eukaryotic cell, this is the nucleus. Right? So, now with the diagram, let's try to understand the containment of genome means a collection of gene means collection of DNA segments. The containment of genome in the nucleus of eukaryote. Now this is eukaryote, right? Or now let's take a prokaryotic cell, a bacterial cell. Now this is the bacterial cell, right? As I initially said, in a bacterial cell, what is a nucleoid? The location where the DNA segments are present in a prokaryotic cell. There the DNA will be present in a circular manner, right? Right. Now what is this? This is a prokaryotic cell. Prokaryote. Prokaryotic cell. What is this? Nucleoid. Nucle. Wait. Nucleoid. What are these? DNA. Circular DNA. Circular DNA. Right now let's from these diagrams let's try to identify or let's try to learn what is the main concept that is being trying to be conveyed, right. The collection of genome, the containment of collection of genome in the nucleus of a eukaryotic cell, or the containment of it in the nucleus of prokaryotes. Either DNA packaging does not speak about only eukaryotes or prokaryotes. They are taking it as a collectiveness. They're saying that the containment of genome in a prokaryote or eukaryote. But if it is in prokaryote, it should be present in nucleoid. If it is in eukaryote, it should present in nucleus. Is it clear? Please drop a message. Is it clear? If it's clear, I'll go further. Clear? Just put a thumbs up. Is it clear? Guys. Because without understanding this, we cannot go further. If it is not clear, just let me know. No issue. I'll again explain it from first. Is it clear? Hello. Right. Thanks. Right. Then let's go to the process. If you understand the definition, half of your problem is gone. Because most of us try to understand the concept without understanding the definition. We are trying to just memorize the definition. So I'll say how to uh, understand the concept from the definition. We'll start from the definition, right? They have given the definition just to start from it, right? Right. Now we'll come to the topic. The first topic, DNA packaging in prokaryotes. Right. Right. Now, now before going to the topic, before going on to the resource book, don't look at the resource book. I'll explain what is the process that is going on. Right. After explanation, I'll give a time, then go on to the resource book. Right. Right. Now, this is what happens. Now we know what type of DNA prokaryotes have circular DNA, right? So the DNA actually looks like this. Right. Now here the bases will be present. Right. What is this? This is the circular DNA, right? This is how a DNA is present in a prokaryotic cell. Now, our education department, for our comfort of drawing, they have given the structure like this. They have given it as just like this, as a circle. Is that clear? They have given it as like this. Because if they question this part in an exam, students should not waste their time in drawing complex structures like circular DNA. So they have given it as a circular structure, circle, right? Now, before starting the diagrammatic explanation, I'll let you know why this DNA packaging happens. There should be reason, right? This is the reason. Now, the DNA that is present in cells, they are not in an organized manner. They are scattered all over. They are located in various locations, right? Now, it is like a country. Now, take a country. If there are no schools, just think of the situation of the citizens of the country, right? Because of the schools that are present, because of the military camps that are present, the individuals that are present in a country are being groomed under a set of rules, right? So similarly, here, those cells that are present in prokaryotes and eukaryotes, they will be trying to arrange the DNA that are present in different manner in a cell to a completely organized manner. That is the main purpose. They're trying to organize the DNA that is present in cells in different locations. The scattered DNA that are present should be arranged in an organized manner, right? That is the main purpose. Now, here the DNA is ready. Now, this is the DNA. Now, before DNA packaging, just think now. Don't take every word that I'm saying to be. Don't expect every word that I'm saying to be in the resource book. For your explanation purpose, I am using some sort of words, right? Now, just think before DNA packaging, the cell gives a signal. No, the resource book they haven't given those. The cell gives a signal. Those stuff they haven't given, right? Now, I'm just saying for explanation. Now, before the DNA packaging, what happens is some sort of complexes code. Now you know, in a cell, DNA is not only the components that are present. There are proteins, there are carbohydrates, there are multiple components that are present in a cell, right? So similarly, here in the cell, there is a complex called core, right? It has a shape like this. Just think, it has a shape like this green parts. What is this? This is the core. Now, the resource book says this core is made up of RNA and protein. Clear? Core is made up of RNA and protein, right? Now, what happens is when the cores that are present, these cores that are present in a cell during the process of DNA packaging, they will be trying to push the DNA, the circular DNA that is present, right? Just think a flexible circular rod, right? Now, when these green colored cores exerts a force towards the center of the DNA, the structure that is looking like would be like this. It would be looking like this. And here, the cores would be present. Do you agree with me? Just think, they're exerting a force. Now here also they'll be exerting a force. Now this part will be getting a curved shape. This will be getting a curved shape. This will be getting curved. Is it clear? Is it clear? Right. Thanks. Right. Now what happens is this process where the cores assemble and help the DNA to form into a separate shape of structure. This process is called, yeah, this process is called coiling. What is it? Coiling, right? This process is called coiling. So this is circular DNA. This is circular DNA, right? After the process of coiling, what happens is the DNA turns to a structure called a loop, right? A loop. Now this is the loop. Now, can you see here? It would be a hole, right? Remember that here at the center there would be a hole, right? Now, what happens is further the loop will be undergoing a process called supercoiling. Supercoiling. At the end of supercoiling, what happens here is the coiled structure will be getting bit shortened and smaller, and each of them will be further coiled like this, right? Still the cores are holding them here at this point, right? At the end of the supercoiling process, the structure that is formed is called a domain. A domain, right? Now, initially, just think in a cell, in the nucleoid, the DNA is present in a prokaryotic cell, which is circular DNA. When the DNA packaging time is ready, the cores that are present, cores, what are cores? Cores are some complexes which are made up of RNA and protein, right? Don't think about those for bit complexly. Just remember, core is a complex made up of RNA and proteins, right? When the DNA packaging is closer, the time is ready, those cores will be coming closer and they will be allowing the DNA, which is a circular shape, to turn its shape from a circular shape into a loop-like shape, like a flower-like shape. That process is called as coiling, right? The circular DNA, after the process of coiling, will turn into a structure called loop, right? That loop will be undergoing a process called supercoiling, and at the end, it will be forming into a structure called the domain, right? Is it clear? This is the process. This is what comes under DNA packaging. This is the entire thing. Is it clear? Please drop a message if it is clear. If it's not clear, please let me know. I'll explain it again. Is it clear? Guys, is it clear? Please drop a message. Okay. After noting it down, drop a message. We won't note it down. Note it down. Shall I move? Let's try to complete it within tonight. So Have you noted down? Right, perfect. Right. Now this is the process. Now let's go to the note. So your resource book introduces that. Now this is the now we are wait a minute. Now resource book note. What your resource book says. Now first thing your resource book says is the DNA packaging process. Packaging process in prokaryote is done with the help of certain proteins, right? Proteins. So, can you understand what are those proteins? There are multiple proteins. Now, they have allocated only this portion of knowledge for your syllabus. So, you don't know. Now, there might be some other proteins too, but for your knowledge, this is what they will be tested, right? So, for our knowledge, we can say it might be core. Core is one of the proteins which helps for, it is a protein complex which helps for the DNA packaging process. Am I right? Right. So, and then the second thing is, right. Second thing, they are saying that initially the circular DNA will be coiled into loops and they'll be further coiled to super coiled to form loop domains, right? We have seen it in the diagram. And then they are saying that cores attaches the chromosome to the membrane. So this is some sort of a point which can confuse most of the students, right? What is the point? They are saying core attaches the chromosome to the membrane. Right. Now, if you break down this point, you can understand that here. Now, what happens here is, if you see, now just take the loop domain here. Now take the loop domain here, right? Now at the end of the first uh, process of DNA packaging, the loop domain. So number one. Now there might be multiple DNA packaging taking place and multiple ones will be getting collected. Am I right? Right? Wait. Right. So those will be getting collected. Now you might feel where does this go? They will be arranged, but how? So another point that bit confusing here is they're bringing the word called chromosome here. So we haven't discussed something about chromosome under the topic of DNA packaging here yet. So what they are telling indirectly is now this is what happens. Now see, wait, I'll draw it in black. Now this is um, the supercoiled DNA that is present, right? This is the supercoiled DNA, right? There will be another supercoiled DNA that can be present, right? This is another supercoiled DNA, right? So here the cores will be present. Don't forget that the cores will be present. Those are the ones that have been helped those DNA to get that shape. Now these are the supercoiled DNA. Now here what happens is now I said you initially, right? Here there is a hole. This point, it is a hole, right? Can you imagine it can be a hole here? If you can accept that the red part that I colored here is a hole, please drop a thumbs up. Oh, just put a message, yes, sir. Can you accept that if those cores change the shape into a loop, that red part would be a hole? Do you agree that? Do you agree? If you can't understand, just drop a message to explain it again. I'll explain. Do you agree? Please drop a message. This part here, this is a circle. Do you agree this is a circle? Right? Now, what happens is like this. Just imagine there is a protein in a cylindrical shape. They haven't given this in your resource book and they haven't given this content also. But for the explanation, I'll give you that knowledge, right? Now, this is a uh, cylindrical shape structure. Now, this is protein. If you be more precise, it is called a protein scaffold. You would come across this word further when you're going. Next, the next part will be this scaffold, right? But they haven't given this word under the DNA packaging in prokaryotes. I should be clear with that. They haven't given the word protein scaffold in under the note of DNA packaging. They have given it under eukaryotes, not under prokaryotes. So, right now, if you take this protein, what happens is these coiled ones, these supercoiled DNAs that are present, will be moving within this. Can you connect it? How would be it looking like? Now, it will be looking like this. Now, if you see, the DNAs that are coiled will be in this manner. The center part will be going across that protein like this. This hole, the red part here, will be going in this manner. It gets collected and gives a bit of fluffy nature. So that is called as a chromosome. This supercoiled DNA and the protein is called the chromosome. So this is what T.H. Morgan said. T.H. Morgan, in his discovery, he said that, what did he say? He said, T.H. Morgan, this is what he said: chromosomes. Chromosomes are made up of two things. What are the two things? I introduced those two at the start of today's class. Can anyone remember? He said that chromosomes are made up of DNA and proteins. That is what he said. He said that they are made up of DNA and proteins. Now, do you agree chromosomes are made up of DNA and proteins? Now, roughly what happens is the DNA that are present in the circular shape will be changed into shape, a loop-like shape with the help of those cores. And those cores will be binding those tightly into loop shape. Those loop shape will be forming a hole-like shape at the center. That process is called as coiling. Those loops will be undergoing supercoiling to form loop domains where they'll be bit more supercoiled, right? No, now those supercoiled DNA that are allocated, those will be separated and those will be getting packed one after other with the help of the protein scaffold and they will be forming a chromosome. Is it clear? Is it clear? If it is clear, please respond. If it is not clear, also just drop a message. You need to understand. Is it clear? Right. No doubts? If there is something that you didn't understand, just point it out. Just point out it. I'll explain it again. Any doubts? All right. Right. A minute. Right. Right. Now, further in the resource book, now I explained two statements. Now, when you now after explaining that, just try to understand that. Can you understand this point? Core attaches the chromosome to the membrane. This core attaches the chromosome to the membrane. Now, cores will be present here. So, cores will be present here. The green parts are the cores, right? Right. Now, here membrane, they are saying instead of using the word protein scaffold, they are saying membrane. Membrane in the surface. Now, just think membrane, they are trying to convey what is it? The surface where it touches the surface where the core touches to the protein is called a membrane, right? That is what they are trying to say. The core attaches the chromosome. Chromosome means the loops. Chromosome to the membrane. Can you understand that? Now this outer layer where the core touches to the protein is the thing they are trying to convey as membrane. Is it clear? Please drop a message if it is clear. That point, the last point, core attaches the chromosome to the membrane. Is it clear? Is it clear? Right. Right. Now that is the explanation for the third point. Now let's go over that was the explanation for the second point. Now it's for third point. Now, third point is they are saying that removal of RNA, removal of RNA leads to the loss of independency of loops. By reading this statement, can you understand what they are trying to convey? They're saying removal of RNA leads to the loss of independence of loop. So where you might ask, so where does this RNA comes here? Now, this is what we learned, right? This is what we learned. Process here the code. That is what they're trying to tell indirectly. Yeah, the code, right? That core is a thing they are saying removal of RNA. Now, instead of RNA, we can think of core. Removal of core leads to the loss of independence of loops. Independence means the shape, yeah, the unique shape that has been obtained after the coiling process can be lost if the core is removed. Correct, right? Now, if you remove that green part, those flower shaped structure will be completely getting back to the initial circular shape or sometimes a shrunken shape, right? So that is the thing they are trying to convey or trying to say from that point. Removal of RNA leads to the loss of independence of loop. Clear, right? That point, right? Clear? Oh, I hope it's clear. Now, the fourth one. We are saying that The supercoiled DNA, the supercoiled DNA can be relaxed by introducing single stranded nicks called DNAs, right? Now, can you see? Now we have obtained completely filed DNA at the end of DNA packaging. Now they are saying that removal or supercoiled DNA can be relaxed by introducing single-stranded nicks. Now, what is this nicks means? It is type of a protein. Just think it is a type of a substance. It's a protein. This, what it does is, can you remember phosphodiester bonds? Can you remember the term phosphodiester bonds, which we came across during the unit 2? So if you see phosphodiester bonds, phosphodiester bonds, those are the bonds present between nucleic acids. Right. It is the bonds that are present between nucleic acids, not between nucleotides. You should not confuse that. The bonds that is present in between nucleic acids is phosphodiester. The bonds that are present in between nucleotides is, what is the bond? Hydrogen bonds. Right? I'll write it down. Hydrogen bonds is the bonds or present between nucleotides. Now if you can't connect nucleic acids nucleotides, simply this is the thing. Now just think, this is the DNA, this is a strand of DNA, right? Now these are the nitrogenous pairs. Now this is adenine, this is guanine, this is cytosine, right? As per the rule of complementary base pair rule, what are the bases that should be present? Here adenine means if it is a DNA, it should be thymine. If it is guanine, should be cytosine. If it is cytosine, it should be guanine. Right. Now, this is the red part is a strand of DNA. This red part is a strand of DNA. Between two strands of DNA, that is between two strands of DNA, nitrogenous base pairs are present. What are nitrogenous base pairs? The blue ones, right? Those nitrogenous bases are based with a type of bond called the hydrogen bond. That is the here like this, right? So that is the thing he mentioned between nucleotides, hydrogen bonds, right? If you say nucleic acids means just I'm not taking about talking about deoxyribonucleotide. Now this is the deoxyribonucleotide. Nucleic acid means DNA. The bonds between DNA, not a strand, between the DNA molecule, between one DNA molecule and another DNA molecule, the bond that is present is phosphodiester. Right? Right. Now here the statement says supercoiled DNA can be relaxed by introducing single-stranded nicks. These DNAs, what they will be doing is they will be breaking the phosphodiester bonds between the nucleic acids. Between nucleic acids. Now, if you see, this is a strand of DNA. This is a strand of DNA, right? This is a strand of another DNA, right? Now the type of bond that is present in between them is phosphodiester bond. So here, single-stranded nicks called DNA. Now this is a phosphodiester bond, right? The single-stranded nicks. I'll give a shape. Now this is the phosphodise single stranded nick called DNase. Now what they will do is they will be coming into this location and they'll be breaking the bonds that are present and they'll be relaxing, right? And if it is relaxed, what happens is now the structure we have obtained here, this part, the domain, the structure we have obtained here will be completely changing its shape into like this now. If relaxing, if relaxing, right, it will be changing its shape to like this, like this. This will be the shape. Roughly, it will be looking like this. The cores will be yet attached, but still it will be eventually the cores also starts to remove because the loops start to get apart from each other, right? That is the thing they're trying to convey. Is it clear? Is this statement clear? I'll give her again an explanation. They're saying supercoiled DNA can be relaxed by introducing single strands called DNase. DNase is a type of substance, a protein, which breaks the phosphodiester bonds between nucleic acids. Right? It will be breaking the bonds between them and it will be relaxing. If you take this structure which I given, the domain, if DNase comes into the domain, they will be relaxing it by breaking the phosphodiester bonds that are present between the loops here, the between the loops, and they will be changing the shape into like this shape. And they will be changing the shape, right? Now you might ask why those DNases want to relax this uh, supercoiled DNA because for DNA packaging, the main requirement is the DNA should be coiled, right? Now, so after so much of hard work, so much of process that went through, why should a supercoiled DNA get relaxed? The reason is this. If in case, now this point is also not mentioned in your resource book, but for the explanation process, to understand why you should have this knowledge. Now, what now let's see why relaxing. Now, if you see initially before before the filing process, the DNA will be circularly, it will be looking like this, right? If you see after, it will be looking like this, right? It will be completely coiled and then cores are attached, right? It will be looking like this. Now, now the initial features that are present here should be present here, right? During the process of coiling, coiling and supercoiling, if there are any errors that have taken place, if there are any mistakes that have taken place, if some features, some information that are present in the initial DNA are lost in the final DNA, it is an useless process, right? It is less efficient. So what it does is the cell itself will start to use the DNA that are present to relax it. Is it clear? Will be using those components to relax it because it is no more use of it because the initial information that are present due to this process has some sort of lost. Now, what we had initially should not be lost, right? If a process is there, right? Now, just think like this. Now, we are doing a business. Initially, we deposit some sort of money and we are getting into it, right? With the time, if we get a some sort of an income, it should cancel off or it should be surpassing the amount we have deposited initially. If so, it is a beneficial one, right? If in case there is a further loss that takes place, it is a less efficient one. Similarly, initially the DNA had some sort of information, some sort of features in it. During this process, if there are any mistakes, there can be errors that can take place, right? So if there are any errors that took place, some information might be lacking in the final structure. So if that is the case, the cell will be using the DNases that are present to relax the DNA that is supercoiled. Is it clear? Now, this is not given explanation in your resource book, but most of the students find it difficult to understand why these statements are given without a proper explanation. Just for an explanation, I gave this idea. Is it clear? Can you understand it? Is it clear? Right. Thank you. Now, clear, right? Now, right. Now, I'll explain four statements. Now, this is the last one, right? Fifth one. Let's go to the fifth, right? I'm picking those important ones, which is difficult for the understanding of students, right? So the fifth point is they're saying that this supercoiling and relaxing is useful during a process. Now, this word is new for you all, transcription. Now, don't wonder what is this transcription, because you will be going across or will be introduced to that when you're in the future, right? Now they're saying that supercoiling and relaxing is useful during transcription. Transcription means, simply for the understanding for the moment, I'll give a rough idea. Transcription means it is a process of protein synthesis, right? Normally, protein synthesis has two sub phases. There are two phases for a protein synthesis to take place, right? First one, transcription, translation. You no need to note it down, because under this part it is not given, right? So when we are studying protein synthesis, let's talk about that. Just for the explanation of this statement, I use that, right? Transcription is one of the phase of protein synthesis, right? So they are saying supercoiling and relaxing is useful during transcription. Now, why they are saying it so is, now if you see, eventually now we are studying DNA packaging with a motive that we have to arrange the DNAs in an organized manner. Now, next topic will be DNA replication, where we'll be studying why that packaged DNA, or for what purpose it will be used. It will be used to produce multiple copies, and then it will be used to produce proteins, right? Main purpose is to produce proteins, right? So they're saying that supercoiling means they should be organized, right? Relaxing means to mean. Now, why should be relaxing useful for transcription means to minimize the error, right? Error. Now you might think, what to minimize error means, what is there? Now, if relaxing is not present, all the DNA with information that has lost, right? Can you understand that point? Just try to understand it. All the DNA, right? All the DNA with some lacking informations also compete for the protein synthesis, right? Not the DNAs that have preserved their features. All DNA. There might be DNA that has lost its features, lost its information structure, right? all will be starting to complete the protein synthesis. So that is not useful because there can be multiple errors taking place. Now, if there is a DNA, now see, for example, if you see, now see, there is a DNA, there is an error, right? There is an error. Now, this DNA, if now assume that there is no relaxing process, right? Now it has been coiled, supercoiled, and after supercoiling, it has produced into a structure. It has produced into a coiled structure completely, right? For the explanation of this. Now this is completely supercoiled. Now, initial there can be now just think there are 100 information initially. Now we are only 75%, right? Now for the protein synthesis, if it undergoes the protein that is produced, right? It might contain 25% of loss in its information, right? Is it clear? Because already some information has lost. During this protein synthesis, there are chances there might be much information to be lost also. So it might be an inefficient process if there is no relaxing involved. So that is what they are saying. Supercoiling and relaxing are useful during transcription, right? Is it clear? That's all for DNA packaging in prokaryotes. Now go through this complete structure and with your note, refer them, compare them, and drop a message if there's a doubt. This is the basic structure for the diagram. This is not given in the resource book, but this is the process that is taking place. Is it clear for all? Because it's an important part. Now we have concluded the DNA packaging prokaryotes. So this can be questioned. So is it clear? I hope it's clear for all. Yes. We were both right? Right. Thank you. Now, that's it for the DNA packaging in prokaryotes. Now, if you understand this, DNA packaging in eukaryotes is nothing. It's a small one, right? Compared to this, it is a small one, right? Let's finish it. And then DNA packaging topic would be, it will be coming to an end, right? Last one, keep that focus that, till then we can quickly finish it, right? Now, DNA packaging in eukaryotes. Right. Now here, here there's a bit different, right? The concept is slightly different, right? Here the DNA packaging in eukaryotes, it happens in 4 levels, right? It takes place in 4 levels. So I'll mention it here. Wait. Right. It will be taking in level 1, level 2, level 3, and level 4. 4 levels will be taking place, right? So here they have given certain names of proteins, they have given certain dimensions. So simple part, but they have given something, some information to remember, right? Now, here also let's go with the definition, right? What is the definition under DNA packaging? It is the containment of genome in nucleoid of prokaryotes or nucleus in eukaryotes. So here, if DNA packaging takes place, it should take place in the nucleus, right? So it should take place, right? First one, nucleus. The process takes place in the nucleus, right? Right. Now here, there were what type of DNA? Circular DNA. Now here, if you see, it's the opposite. They contain linear DNA. Linear DNA is present in nucleus, right? Now you should not come to a conclusion that eukaryotes have only linear DNA, prokaryotes have only circular DNA. No, prokaryotes have only circular DNA. That's right. But eukaryotes have both linear and circular DNA. You should remember that, right? I'll just revise that also because it was given under Unit 3. If you can't remember that, I'll revise that. Eukaryotes have two types, linear and circular, right? This linear is present in mainly nucleus, right? Nucleus. This circular is not much considered by us, but it is also present. Circular is present in mitochondria. Can you remember? Mitochondria, mitochondrial matrix. It is present in mitochondrial matrix and is present in stroma. Stroma of chloroplast. Chloroplast, right? So now say, does eukaryotes have only linear DNA? No, they have both linear and circular DNA. But prokaryotes have only circular DNA, right? Right. Right. Now I'll explain the process and then let's go to the resource book comparison. Right. Now here what happens is, now here what happens is Now here what happens is it takes place in 4 levels, as I said. So let's go one by one after each level. Right. Now the first level. Right. In level 1, what happens is here, not like prokaryotes. In prokaryotes, even the ones who made the book, the resource book, they have given the information as a complete pack. But here, for each level, they have given separately the information and they have given what the process that is taking place completely and separately. Each and everything they have given. So here, what happens is like this. Now, there are 8 proteins that are arranged like this. One, two, three, four, and there will be four at the back, like this. Right. Now here, this protein is called as one protein is called as histone. Histone protein. Can you remember? This was given under Unit 3. So in the level 1 of DNA packaging, what happens is the DNA that is present, that DNA will be winding around the complex like this. DNA will be winding, completely winding like this. Right? Completely winding like this. Right? This complex after completely winding. Completely means don't think it will be winding completely to cover the histone protease. No, it will be just winding it. Now, like this, wait. Now it will be like, actually, it will be taking like this. These are the histone proteins. 8 proteins that are present. Right now, and then the DNA will be present in this manner. It will be completely winding and it will be right. It will be like this. So, what is this red color circle-like structure? It is histone protein. How many histone proteins are present? 8. Right. Now, what is this blue structure? Blue structure is DNA strand. DNA strand. Right now, this complete complex with 8 histone proteins and a DNA winding it, this complete structure is called as a nucleosome. Right? This is what happens in level 1. That's it. Now let's go to the level 2. So simple, just few information that is given. So we can quickly finish it off. So level 2, what happens is, now this nucleosome is present, right? This nucleosome, it has a dimension. Right here, I'll mention it. The nucleosome has a dimension of 10 nanometer. Right? This nucleosome will be packing. Now here, in the second level, they have given certain terms. Right? The they are saying 10 nanometer fiber twists and packs in a spiral fashion. Now you might think, where is the fiber? So fiber means the nucleosome. Now nucleosome, these have small dimensions, right? Nanometer structures. 10 nanometer means is a very small structure, right? So they are just assuming it to be a fiber-like stretch, a fiber stretch, fiber-like structure, right? So they are saying the 10 nanometer fiber twists, it will be getting twisted and packs in a spiral fashion. So one nucleosome, one fiber will be getting completely collected, and that will be forming 10 nanometer fiber twists and packs in a spiral fashion to form a 30 nanometer fiber. Right? Simply, what happens is, now instead of drawing the 8 histone proteins, I'll draw it like this, right? A complete circle subdivided into 4, and at the back there are 4, right? Assume like this, right? It'll be easy. Now, this, now can you say what is the dimension of this? 10 nanometer, right? Now it will be getting collected, packed in, like it will be getting packed in this manner, looking like this. There will be multiple ones, right? Multiple ones will be present, and there will be DNA strands too, right? Like it will be like this. So this has a diameter of uh 30 nanometer, right? 30 30 nanometer, right? That's it. So just think like this. Can you connect it with something like marble stripped with some sort of a ribbon, and those marbles are connected and packed into form a complete separate structure, a complex, multiple marble complex, right? Something like that. Now, here something, a new thing they have been trying to convey here. Yes, I have heard it right. Chromatin fiber. And chromatin fiber, you learned this under cell division, right? So chromatin fiber is made up of DNA and protein. Do you agree with me? DNA and protein. Now see here, the red part is the protein and the blue part is the DNA, right? So chromatin fibers are made up of DNA and protein, right? Is it clear? Is it clear? Now this is the chromatin fiber, the 30 nanometer one, not this. 10 nanometers a nucleosome, right? Will be getting collected to form a 30 nanometer fiber. So this is the one that is called as a chromatin fiber. Is it clear? If it is clear, quickly mention it in the chat. Chromatin, chromatin fiber. Is it clear? Yeah, I hope it's clear. Right. Now let's go to the level 3. Now, level 3, what happens is now instead of the complete chromatin fiber, instead of drawing those marble like structures and blue, can you think of it as a complete stretch, something like a stretch of a structure? Now like this. Now it is looking like this, right? It is looking like this, right? Instead of this complete structure, these are nanometer structures, right? 30 nanometer. If you see it in a microscopic level, sometimes you might see it as like stretch like structures. Can you stretch stretch like structure? Can you agree with that? Now, even if a camera, the camera, the more you go in zoom, the structures are more delicate and they are more precise, right? Where when we are coming more far from the location, those structures look like simple structures. Do you agree with me? So similarly here, if you see it microscopically, right, more zoom in, it will be looking like this structure. But if you see it as an overall, it might feel like a strip like fiber like structure. It's a fiber, right? So fiber like this back like structure. Do you agree with that? If you agree, please drop a message and I can continue. Is it clear? Is it clear? Is it clear, guys? Right. Thanks. Right. Right. Now, you agree with that, right? Right. Then, if you agree with that, you can understand this. Now, here what happens is, uh, wait, is it now? Just now, I'm taking instead of this complete structure, I'm taking it as a strip, right? Now, here a protein called, I introduced this initially, protein scaffold. Can you remember? Protein scaffold, right? This protein will be present and the fibers, the chromatin fibers that are formed, they will be forming radial loops like this. These black ones are chromatin fibers. They'll be forming radial loops. Radial mean up and down, loop, radial loop, and this is chromatin. Can you understand that? Is it clear? They're forming radial loops, right? They're forming radial loops, and it has a thickness. Now, this structure has a thickness of 300 nanometer, right? It has a thickness of 300 nanometer. Is it clear? Right? Now, here, the now chromatin fibers are the ones that forms the loop, radial loops, right? Now, these loops that are formed, the collection of loops, are called the loop domains. Can you understand? Loop domain. And this is a loop collection, right? So loop domains. Wait a minute. Loop domain 300 nanometer thickness, right? That's it. Now level 4. Level 4 is easy. Level 4, what happens here is straight one structure, right? Actually, they will be looking in a completely loop-like structure. Actually, they'll be looking like this, right? And then, as we studied in the level 3, the loops will be in this manner. Loops of chromatin fiber, the loop domains, right? Loop domains will be in this manner, right? And here they are resembling the chromosome. The mitotic chromosome will be formed, right? If you see the resource book, you can understand from the picture itself, because the picture gives the clear explanation. Yes, centromere. And if you take the black, is it clear? It is looking like this, right? Here also they are using certain words. Now, under the level 2, I use certain words like twists and packs in a spiral fashion. So here also they're using the loop domain, coils, poles, and further compacts. Right? 3 words: coils, poles, further compacts. Coils, poles, further compacts. Is it clear? Now I'll give just a rough idea. A minute. Right. Right. Let's go. Right. DNA packaging in eukaryotes. Eukaryotic DNA packaging means containment of genome in the nucleus of eukaryotes. Right. It is associated with the linear DNA, linear type of DNA in eukaryotes. Right. Eukaryotic DNA packaging takes place in 4 levels: level 1, level 2, level 3, level 4. Right. Here also it takes place with the contribution of Wait. Sorry. Contribution of proteins. You agree with me, right? Proteins, right? Here, the first level, what happens is a complex of 8 histone proteins are being built in a shape of structure, and the DNA will be winding around it, forming a structure called a nucleosome. This is the nucleosome. Now, here, the nucleosome, now see, this is the one nucleosome, this is another nucleosome. Now, between two nucleosomes, there would be a stretch of DNA which connects the two nucleosomes. Now, this is one nucleosome, this is another nucleosome. The stretch of DNA that connects the two nucleosomes, this is called as linker DNA. Can you understand that? Now, like this. Now, this is the nucleosome. Sorry, this is a complex of histone proteins. Right here, it is another complex of histone proteins. Right here, the DNA will be present here. It is completely winding this. Right here, there is another. Right now, between these two, the connecting one between these two, this one is called a linker DNA. Linker DNA. Clear? This is called as a linker DNA. Now this complex, yeah. This is a nucleosome, right? Right. Now, if you to go to the level 2, what happens in the level 2 is the 10 nanometer nucleosome that is formed, or the 10 nanometer complex will be twisting and packing in a spiral fashion to form a structure called the chromatin fiber, which is 30 nanometer in dimension. Right? Right. This is the structure that is given. And in level 3, what happens is the chromatin fiber that is formed will be attaching to the 30 nanometer fiber that is formed will be attaching to the protein. What is the protein? Protein scaffold. And it forms loop-like structures called the loop domain. The thickness of it is 300 nanometer. Right? In the fourth level, what happens is the loop domains, three things happens. It will be coiling, it will be folding, it will be further compacting to form the mitotic chromosome. Right? And they have said the sister chromatids will be already replicated. You know where they are replicated, right? Sister chromatids are replicated in the S phase, right? And they are saying metaphase chromosome. Now, the diameter of the metaphase chromosome is 700 nanometer. So I'll mention that one also here. Yeah. Diameter of metaphase. Now, this is not connected here. Metaphase chromosome is 700 nanometer. They are given it in the resource book, but you can connect this with the unit 2 cell division knowledge 2. Right? Right. Right. This is the complete thing. Now, last and final part, and then DNA packaging is done. Right? The chromatin fiber, that thing which we learned today, chromatin fiber can be divided into two. First one, euchromatin. Second thing is heterochromatin, which means mainly there are certain features. Depending on the features, chromatin fibers can be divided into two. Right. Now I'll mention what are the features. First one, euchromatin is lightly packed. Opposite of it, heterochromatin, tightly packed, of course. Euchromatin, active in transcription. Again, transcription comes under protein synthesis. This is inactive in transcription. In transcription. Euchromatin, rich in genes. Heterochromatin contains nucleotide sequences. Right? Now, don't ask why the nucleotide sequences are present. They are given it under the note nucleotide sequences are present. And they are given why those nucleotide sequences are present also. They are given three functions. Most probably, if they ask why those nucleotide sequences, while those nucleotide sequences are present, these are three functions you can mention, right? What are three functions? First one, gene regulation. No need more detail about these functions. Just if you can, with the practice of memorization, you can just note it down. Gene regulation, epigenetic, epigenetic inheritance. Examiners won't be asking what is gene regulation, what is epigenetic inheritance. They would be asking what are the functions. So just generally, you can mention the functions. Epigenetic inheritance, and last one, protection of chromosomal integrity. Integrity. Right? Right. Again, chromatin fibers, structure they can be divided into two. First one euchromatin, second one heterochromatin. Euchromatin are lightly packed, heterochromatin tightly packed. Euchromatin is rich in genes. Heterochromatin contains few nuclear sequences. What are the functions of them? Gene regulation, epigenetic inheritance, protection of chromosomal integrity. And euchromatin contains, they are active in transcription. Heterochromatin inactive in transcription. That's all. Right? If they are asked to differentiate under structure essay, you can mention these. Right. That's it. DNA packaging. So finally, I'll give a learn today. Right. Right. First of all, what was the discovery of DNA's shape? DNA is a double stranded molecule which was proposed by two scientists, James Watson and Francis Crick. They proposed them using some sort of technology called X-ray crystallography, which was proposed by Rosalind Franklin. A scientist called T.H. Morgan and his companions, they suggested that chromosomes are made up of DNA and proteins. Right? Under the topic of DNA packaging, we learned how chromosomes are made up of DNA and proteins. Right? Under architecture of chromosome, we know how chromosomes are arranged. Right? Here in this lesson, more commonly, DNAs, they are saying DNA of prokaryotes and eukaryotes are commonly said to be chromosome. But true chromosome, note it down, true chromosomes are present only in eukaryotes. True chromosomes are present only in eukaryotes. Right? Right. Now comes to the DNA packaging. DNA packaging means the containment of genome in nucleoid of prokaryotes and or nucleus of eukaryotes. Right. DNA packaging in prokaryotes. Here, proteins are involved. Right. Those proteins are involved, and initially there's a circular DNA. This circular DNA can be coiled and further supercoiled to form a completely package. It will supercoiled to form a loop. Initially, it will be coiled to form a loop, and then it will be further supercoiled to form loop domain. Here, the cores are made up of proteins and RNA. Those cores will be attaching the chromosome to the membrane. Cores acts as barrier for the rotation. Acts as barrier for the rotation means they will be preventing from any movements. Right? Acts as a barrier for the rotation. The supercoiled DNA can be relaxed by introducing a single strand Higgs called DNase. What will they be doing? They'll be breaking the phosphodiester bonds that are present between nucleic acids. Right? Removal of RNA leads to the loss of independence of loops. RNA means cores. Removal of cores will be completely changing. It will be denaturating the shape of the loop domains that are present. Right? And then we come to the eukaryotic DNA packaging. Eukaryotic DNA packaging is involved with certain proteins, especially histone proteins. Right? Chromatin fibers means DNA and protein complex. So if you see, T.H. Morgan said that chromosomes are made up of DNA and proteins, right? Chromatin fibers are also made up of DNA and proteins, right? Because we know from knowledge of Unit 2, chromatin fibers are the ones that will be condensing and forming chromosomes, right? Right, right. And then the eukaryotic DNA packaging takes place in four levels. Where do they take? They take place in the nucleus. Prokaryotic DNA packaging in nucleoid, right? Four levels. First level, what happens is a complex of 8 histone proteins are arranged. Those will be winded by a DNA strip of DNA, strand of DNA. This complex is called as the nucleosome. It contains 8 histone proteins, right? In the second level, what happens is the 10 nanometer fiber or 10 nanometer nucleosome will be twisting and packing in a spiral fashion to form a fiber called the chromatin fiber, which has a diameter of 30 nanometer. Between two nucleosomes, what is the type of DNA that connects between them? a stretch of DNA, which is called as linked DNA, right? And especially, they are giving a type of synonym. They're comparing the shape of a nucleosome to a bead of a necklace. Can you think of a bead of a necklace? Those circle-like structures, bead of a necklace. All right. In level 2, packs to form a chromatin fiber. Level 3, the chromatin fiber that is forms loops called loop domains attached to the protein scaffold. This is the protein scaffold. Where this is the protein scaffold, right? The red part, protein scaffold. Those are radial loops. They have a thickness of 300 nanometer, right? In the fourth level, the loop domains that are formed happen three things. They'll be undergoing three things. What are they? They'll be coiling, folding, and further compacting to form the mitotic chromosome. The sister chromatids of the chromosomes are already replicated in the S phase, and these metaphase sister chromatids have a diameter of 700 nanometer, right? Chromatin fibers can be divided into two. What are the two types? Euchromatin, heterochromatin. Euchromatin is lightly packed, heterochromatin tightly packed. Euchromatin rich in genes. Heterochromatin contains nucleotide sequences. Nucleotide sequences, it has three functions: gene regulation, epigenetic inheritance, protection of chromosomal integrity. Euchromatin is active in transcription. Heterochromatin is inactive in transcription. Right? That's it. So go through this, go through the note that is given under the resource book, and let me know the lesson for the today. Is it clear? Right? The topic DNA packaging. Is it clear? I have completely summarized or explained the part of DNA packaging. Is it clear? One student has mentioned it is clear. I need the confirmation from the other student also. Is it clear? If it's clear, then we can wind up the class for the today. It's not clear, just let me know. I can explain it again. Is it clear? Yeah, right. And then that's it for the day. Thank you for joining. Hope you see. Right. Thanks for joining. Bye.